in this talk, I will talk about two applications of statistical models to single-cell RNA-seq data analysis, both of which are recent works in my group. And so first of all, from this figure from Wikipedia, we see how the single-cell RNA-seq experiment is done. So basically, there needs to be a dissociation step to obtain individual cells. And we need to be able to label the RNA within individual cells. And each RNA molecule will be reversely transcribed as cDNA and going through some amplification steps such as PCR and then get sequenced. So compared to bulk RNA-seq, the key difference here is that we are able to label the sequence reads for each cell. After pre-processing, our data is in this tabular format, which have cells as rows or cells as columns, and then these correspondingly the columns or the rows will be genes. So it's a cell by gene matrix or a gene by cell matrix, and entries are counts. So we often start from here. And given the single-cell RNA-seq data, many analyses can be performed. And I'll give an introduction in the second part of this talk. And first of all, I want to say that in this talk, I will talk about two recent work. PseudotimeDE, it's a method for inferring differential gene expression along cell pseudotime. And the key is that we want to emphasize the validity of p-values for this inference. And this work was done by my second-year bioinformatics PhD student, Dongyan Song, and it's currently in press at Genome Biology. And this is the bioRxiv link. I think Shuram sent this previously, if you want to check it out. And for the second part, I want to talk about a simulator called scDesign2, which is supposed to generate realistic single-cell gene expression count data. And the key is to capture gene correlations. And this work was done by my PhD student, Tianyi Sun, who is a fourth-year statistics PhD student, and he's also here to join the discussion today. And this work has been accepted by RECOM and is currently under revision at Genome Biology. And you can check the bioRxiv version of this paper. So I'll talk about PseudotimeDE first and scDesign2. And also, since this is a lecture, I'll give some references for further reading if you're interested in some of the techniques. So first of all, what problem does PseudotimeDE aims to address? It is in the context of analysis called pseudotime inference, also known as trajectory inference, a very important analysis in single-cell RNA-seq. So then first, what is pseudotime? It is a latent temporal variable. I quote temporal because it's not really time, but it's something we infer as time from the single cell data. So this variable gives one cell a value, and it's usually normalized between 0 and 1. 0 means the least developed cell, and 1 means the most developed cell. So it's a way to reflect a cell's relative transcriptome status among all cells. In other words, pseudotime inference is trying to order cells along a trajectory or a lineage based on transcriptome similarities. Of course, it may infer that there are multiple lineages, but within each lineage, each cell will receive, cells in that lineage will receive a time, and that's called pseudotime. And like I said, it's usually normalized to 0 to 1. There are several popular methods for pseudotime inference, including Monocle 3, T-scan, and slingshot. Okay, so this is a figure that illustrates the idea of slingshot. Sorry, there's a typo, it should be slingshot. So what it does is that first it performs clustering, and this is a two-dimensional visualization of the cells. You can consider this to be PCA. And then given the clusters, it uses the minimum spanning tree to connect the clusters. And then it tried to smooth it out by using the principal curves. Okay, this is the principal curves. And then it will project each cell to each principal curve. And then after the projection, the coordinates in each curve are considered to be the pseudotime in that lineage. So each curve represents one lineage, and you're going to obtain the pseudotime of cells in that lineage by projection. So this is just a bifurcation trajectory. And here we have a more complicated trifurcation trajectory. Okay, so then for biologists, given the pseudotime or cell trajectories, a very important question is what genes are differentially expressed along each trajectory? And we call these differentially expressed genes short as DE genes. So its formal definition is based on statistical hypothesis testing. That is, if a gene has its expected expression changes along cell pseudotime, we call it DE. Otherwise, we call it non-DE. So here are two examples. So the CCL5 is very well known to be cell cycle related and also immune response related. So we see that if this is the pseudotime x-axis, y-axis is the log transform count. You can consider this to be gene expression level. And you will see that in the earlier stage in the pseudotime, the gene expression is more lowly expressed, but in the later stage, it's more highly expressed. This fits our definition of a DE. Although we didn't do a statistical test yet, but we can see that visually it makes sense to be called DE. And the other gene, if you look at this expression, it's not changing so much along pseudotime. So we want to call this non-DE. Then the question is how to identify genes as DE or non-DE. So I think there is a chat. How do we know the direction of the DE change? So we don't know. And when we talk about DE gene detection, we didn't say whether it's monotone increasing, monotone decreasing. This is something we're just going to detect changes. And after we call DE, it's the user's visualization to decide, okay, what the change directions are. But throughout this talk, our method doesn't focus on detecting a specific type of DE. We are just testing against the null hypothesis that it's not DE. And this is the usual framework for all the DE analysis in genomics, in RNA-seq analysis, that's not specific to the pseudotime DE, but also like if you want to detect DE genes between two cell types, then people are talking about two-sided tests. So either cell type could have the gene more highly expressed. So before our work, there are two, I would say, well-known existing methods for tackling this problem. One is called trait-seq. So this method tests if a gene is DE based on a generalized additive model to capture the relationship between a gene's expression and the gene's pseudotime. So we'll talk about this later. And another method is based in the package called Monaco 3. So Monaco 3 can do a lot of things, and Monaco is the first method that does pseudotime inference, but later it incorporates other functionalities. So for example, it has a functionality to test if a gene is DE, and it uses the generalized linear model, not the generalized additive model. So I'll talk about their differences. And both methods are regression-based. So in the regression, the response is a gene's expression level in a cell. And the predictor or covariate or feature is the cell's pseudotime. So you want to predict from pseudotime about expression. And then you want to test the corresponding effect size of the pseudotime in the regression model, whether that is 0. And by testing it 0 or not, you're basically testing the gene being not DE or not. But in those two methods, there's a common limitation. That is, both of them treat cell pseudotime as a fixed covariate and ignores the uncertainty of pseudotime inference. But pseudotime is actually random. Why? Because we do not observe it as a characteristic of a cell. Instead, we inferred it from the all cells gene expression levels, all gene expression levels. So this inference step itself would involve some uncertainty. And the degree of uncertainty depends on the pseudotime inference method. So our argument is that this ignorance of pseudotime uncertainty may result in invalid p-values. So we'll see that, we'll check that and see whether this argument makes sense. Okay, so to show you that pseudotime inference uncertainty exists, sorry, I should correct this to be pseudotime inference uncertainty, not our method uncertainty. This is a typo. So what this means is that here we're showing, okay, so this is slingshot, one method, and this is the monocle 3, another method. So we're just following each method's practice. So in slingshot, it recommends users to do PCA and then to fit the trajectory or lineage in the PCA. So we see it. And how do we evaluate the uncertainty? What we did is that we perform subsampling on all cells. In each subsample, we just sample without replacement 80% of the cells. And based on that subsample, we apply slingshot. So as a result, every cell will be approximately included in 80% of the subsamples. And so here the y-axis shows the cell's pseudotime in the full sample, and the y-axis shows each cell's pseudotime in the corresponding subsample, if that subsample includes the cell. So then we can see that so the cells are ordered vertically by their pseudotime in the full sample. We can see that each cell has some uncertainty for its pseudotime in the subsample. So the width of the band is actually indicating the degree of uncertainty. This is for slingshot. And this is for monocle 3. As recommended, monocle 3 asks users to use the UMAP for dimension reduction. And this is its trajectory. So for each cell, at least for this particular simulated data set, we can see, yeah, it's actually having a greater degree of uncertainty. And this is another example where we have a bifurcating trajectory in the full sample. And then in the subsamples, sometimes the inferred trajectory is not bifurcation. So these are marked as F. You see that we only capture part of the bifurcation trajectory. So these examples illustrate, yeah, indeed, pseudotime inference has uncertainty. So to address this issue, we propose.
Jessica.
Yeah.
So typically, these pseudotime inference methods, they first perform some kind of dimensionality reduction.
Yeah.
Then they use the reduced representation of the data to build their pseudotime trajectories.
Exactly.
I see. So that's kind of the common workflow of methods, rather than work on the original feature space.
Right. Yeah. They try to get rid of the high dimensionality by dimension reduction first.
I see. And then they fix that, and then they perform the rest of the downstream analyses.
Exactly. Yeah. Thank you. Yeah. So this is our proposed method, PseudotimeDE. And here is the illustration. So we try to capture the uncertainty in pseudotime inference via subsampling. So let's first look at the original data. So this is the whole sample. We perform pseudotime inference, and then we will apply, which I'll introduce later, negative binomial generalized additive model or zero inflated negative binomial generalized additive model, short as GAM, to each gene J, and to calculate a test statistic SJ. Now, please note that we're not going to use the theoretical distribution of the test statistic SJ, the asymptotic distribution, as in the GAM theory, because here, for our motivation, the pseudotime is not fixed. Okay, so we will stop here. We'll calculate this test statistic on our original sample. And then for the null cases, to get the null distribution for a test statistic, our approach is that we will do subsampling of the cells to create subsamples. So you may ask why we don't use bootstrap. The reason is that if we do bootstrap, then we will have repeated cells in each bootstrap sample. And those repeated cells will cause issues for some pseudotime inference method. So they may not allow the same cell to appear twice. So that's why, to avoid that issue, we use subsampling instead of bootstrap. And we set the subsample proportion to be 80%, which seems to be reasonable, and we check the robustness for that. So then on each subsample, we perform pseudotime inference to obtain the pseudotime, and to make sure that for these subsamples, there's no association between pseudotime and gene expression. We permuted the cells within each pseudotime. So after the permutation, we basically disrupt the association between pseudotime and gene expression. And then we fit the same model we use here to each subsample. So on each subsample, then we're going to observe a value for the test statistic from those null subsamples to obtain empirical null distribution of the test statistic. So putting our observed statistic value from the whole sample to the null distribution, we can compute the p-value. So essentially, it's basically a subsampling and permutation-based way to obtain a null distribution, to approximate the null distribution.
And
okay, so
excuse me. Could you go over why you permute again? I'm a little confused on that part.
Because
still.
otherwise, if we don't permute, we cannot call a null distribution. Null distribution means that when the null hypothesis holds, what values my statistic would take, right? So to create the null cases, there must be no association between pseudotime and gene expression.
So what's the reason of doing permutation on the subsample data rather than the original data?
Because if we do on the original data, we are essentially treating the pseudotime inferred for those say n cells as fixed. Because no matter how you permute it, you're still permuting the n pseudotime values you calculate here. So you didn't capture the uncertainty in the pseudotime inference. But if you do the subsampling and pseudotime inference again for each subsample, you are basically applying the whole procedure, pseudotime inference and the NB-GAM, and then test statistic calculation. So you included the pseudotime inference as a step in the whole procedure. But if you just permute the pseudotime here, you are fixing the pseudotime.
Yeah, that makes sense.
And so is this, can I think of this as essentially doing, instead of a bootstrap, a jackknife followed by permutation?
Exactly. Yeah, it's like a jackknife. Yeah. Yeah, it's just that I think traditionally people say jackknife is leave one out, but here we are actually just getting 80%.
Right, I see.
Yeah.
Is there any reason behind choosing 80%?
I think our rationale is just like, why would you choose 5-fold cross-validation? If you do 5-fold cross-validation, 4 folds, that's 80% are used for training, 1% for 1-fold for validation. So I think our 80% is kind of like a, I would say rule of thumb choice. But as the reviewer asked, we just check the effect of this percentage on the I would say on the result, and we actually show that 80% is a reasonable choice, but actually if you go to 90%, it doesn't seem to be too bad a choice.
And what was the effect of changing the percentage on the result?
So we also argue that it's pretty robust, but of course you can see that if the percentage is too low, like 10%, then the pseudotime inference will be too random. So the inference, the uncertainty of pseudotime inference will not be really close to the uncertainty on the original sample. So that's why you don't want the percentage to be too small. But if it's too big, then basically you're using the original data for pseudotime inference. So as long as the inference method random seed is fixed, you should expect to get the same pseudotime for every cell. So then it doesn't make sense to do a self-sampling. So there must be some trade-off in the middle. I think it's just similar to the cross-validation, if you think about that problem. So in the cross-validation, when you actually calculate the validation error in each fold, you are only using k-1 folds to train your algorithm. But ultimately, you want to use all the data to train your algorithm. So there is a gap between the training in cross-validation and the actual training. But if you use, so basically you want that k-1 to be large, right? In that sense. But if you want to talk about the validation error part, if your k is too big, then 1 fold is a small percentage, then that validation error will have a big variance in itself. So that's why for cross-validation, there's also a bias variance trade-off. I think the same idea actually applies here.
That makes sense. Thank you so much.
Can you hear me?
Yeah.
Oh, sorry. Sorry, I just want to add some results about this. So after we try different proportion, it seems like if you use a too high proportion, for example, 90%, then you will have a slightly higher power, but your FDR control will be worse. I think it's caused by your p-value will be more far away from the correct p-values. But if you use like very low proportion, for example, only 50%, then your p-value is roughly okay, but your power will be lower. So after we try different proportion, it seems like 80% is a good balance between these two trains. So that's information. Thanks.
Yeah, but I would not make too much in interpretation into that because we only calculate one realization of FDR. We are calculating FDR, not FDR. FDR is actually over many repetitions. Yeah.
Yes.
So I still want to say that it's not really sensitive to 80%.
Oh yeah. In general, it's very robust.
Yeah.
Yeah, I think the connection to cross-validation is particularly useful to make for people who are more familiar with the supervised learning framework. So yeah, it seems like this is kind of the same kind of trade-off.
I think kind of, yeah, exactly. Exactly, because ultimately we want to get another independent sample with the same number of cells to perform pseudotime inference, but we cannot, right? We only have one sample. So we just have to try to use the idea of jackknife to create the subsamples. But still, just as Dongyan said, if you have so basically we want to capture the degree of uncertainty in some realistic way. If the percentage is too small, then we may be exaggerating the uncertainty and that will hurt the power. But if we are just subsampling too many, right, then it's just very close to the original sample, then you miss a lot of the uncertainty because essentially you are not really making this like an independent sample, I would say. Okay, yeah, so I think people have asked all the technical questions, but I just intended to make this as a diagram because I have the details afterwards. Okay, so here are the notations. So I just want to say that please, for the two parts, okay, for this part, the PseudotimeDE, and the next part, scDesign2, the matrix notations are different. So just bear in mind that this is the notation just for this part. So we are going to start with the count matrix. Okay, so we have n cells as rows, n genes as columns, and we denote Yij as the count of gene J in cell I. Okay, so we have n cells. If we perform pseudotime inference, we will get one pseudotime per cell, T1 to Tn, and Ti is the pseudotime of cell I. So it's often normalized to 0 to 1. Okay, to capture the uncertainty of pseudotime T, we subsample 80% cells in Y, so 80% of the rows in Y, and in the B subsample, this is the subsample matrix. Okay, so we contain 80% of the rows. And then we apply pseudotime inference to this subsample. And we denote the corresponding, the resulting pseudotime as T1b to Tn prime b. So n prime is the subsample cell number. Okay, and then we permute those subsample pseudotime. So we call those T1 star b to Tn prime star b. So star means permuted. All right. So then how do we infer the relationship between pseudotime and cell count? So this is the generalized additive model, again, based on negative binomial. And this is the model used in TradeC, the existing method published in 2020. So the model says, okay, for the response Yij, we assume it to follow a negative binomial with its own mean mu Ij. And a gene, this is a gene-specific dispersion parameter phi j. So for this parameter, we don't think it depends on the cell I, but just gene j. But mu Ij depends on cell I and gene j. So to capture the relationship with pseudotime, we're essentially assuming that law of transfer mu Ij is a function of pseudotime Ti. And this function Fj is considered to be additive nonlinear functions. So this is what we call the additive model part. All right, so you see that, so specifically, you can consider HBK to be a nonlinear transformation of pseudotime TI, and we take a sum, weighted sum. So in the GAM literature, this is called the spline function, and here we use a cubic spline function. So we can consider this capital K to be called K naught in the spline function. So this K, the greater the K, the more complex this function is. So the more complicated relationship you can capture. So we just set this K, big K to be 6, because we think this is enough to capture most of the interpretive relationship. Okay, this is the NB-GAM. So related to this, if we talk about the GLM, generalized linear model used in Monaco 3, what's different is the second line. So we're going to have this FJTI to be a linear function, or to be just I will say beta J, like you can cut this beta J times TI. So you will replace this nonlinear part by the linear function. So then you can see that GAM can capture more complicated relationships that GLM may miss. So similar to NB-GAM, if we consider possible zero inflation in our gene count, we can change this to be zero inflated NB-GAM called ZINB-GAM. So here we're going to have a latent variable ZIJ to indicate whether this gene is on or off. And the probability of being on is PIJ. We follow Bernoulli to draw ZIJ. And then given that ZIJ is 1, the gene's on, the count is going to be drawn from the negative binomial with the gene and cell specific mean and the gene specific dispersion. If ZIJ is 0, YJ is going to be 0. So that causes the zero inflation. So then again, we assume that the mean, the NB mean on the log scale is this function, generalized additive function of pseudotime TI. And for the proportion of being on, we consider to be a logic transformation, just as we have in the logistic regression formulation. But to actually make the problem simpler, right? We don't actually assume, okay, this logic has another nonlinear function itself. We just consider to be some linear transformation of the nonlinear function for log Wij. So that for doing that, we don't need to have another spline function. Otherwise, the problem may be a little too complicated. So these are the model assumptions for GAM, NB-GAM, and ZINB-GAM. So then what we're going to do is to fit each model to a gene. And here we're showing 6 example genes. So you see that the blue line is the fit for NB-GAM, and the red line is the fit for ZINB-GAM. You can see the difference. So essentially, when we look at ZINB-GAM, we are actually considering some of the zeros to be not from the NB. So we're not going to use those zeros to estimate the parameters of NB. So what we're plotting here is actually how the log mu ij changes the fitted, the estimated log mu ij changes with pseudotime. So you see that, yes, indeed, we see the ZINB is not so affected by the presence of zeros, because clearly there are more zeros, but the ZINB only goes down a little. While if you look at the NB, it goes down a lot because it's shown that trend. So actually, since you can see, the data is actually pretty noisy for every gene across the cells. So in our package, we leave both options for user's choice. And our advice is that we would have users should examine the data, the gene expression versus pseudotime, and see whether they want to think of the zeros being biologically meaningful. If they think, yes, the zeros are meaningful, for example, here the zeros indeed indicate lower expression, then they should use NB-GAM. But if they think some of the zeros are totally meaningless, they don't want to trust the zeros, then they could use the ZINB-GAM. So we leave this to be users to be a subjective choice, because I think in the field, there's still a very intense debate about whether zero inflation is something biological or just technical artifacts.
So I have a question here. Since the zero inflated model is a generalization of the NB model, shouldn't that, can't we do a formal test and see whether the zero-inflated model is a better
Yes,
fit?
exactly. So if the users don't provide an option for either choice, our default is to fit both and then use AIC to choose. So, but we know that the ZINB is actually more complicated. So our cutoff for choosing ZINB is that if its AIC is greater than the AIC of NB-GAM by 10. So it's just a rule of thumb. We use that to make a choice between the two models. Yeah. Thank you for the question. Okay, so now it comes to the stage of a statistical test. So the null hypothesis using the GAM formulation is that the fj function is a zero function. So if you recall, fj is here. So zero function means that ti has no effect on the log byj at all. So that's the null hypothesis, and the alternative is that fj is not zero. So our approach is that we first fit a GAM to the count matrix and the infer pseudotime y and t. And we obtain estimates of this function fj applied to the n pseudotime t1 to tn. So this is the n-dimensional vector. We obtain an estimate for it. We call that fj hat, both phase for the vector. And correspondingly, we are going to estimate the covariance matrix of this n-dimensional vector. So this is going to be an n by n matrix. And we denote that by v hat fj. Okay, so based on fj hat and this estimated covariance matrix, we're going to obtain this sandwich-like statistic, right? So basically, we multiply fj hat transpose times the rank R. So I should say this is the rank R pseudo inverse of this covariance matrix, estimated covariance matrix, and then fj hat again. So this will give me a scalar test statistic value. So in the GAM theory, there is derivation for the asymptotic distribution of this test statistic, which is a chi-square distribution or a mixture of chi-square distribution. And its degree of freedom will depend on the complexity of the fj, the number of knots. And that actually, the Tracy paper, they also use GAM, and they actually use the asymptotic distribution to calculate the p-value. But as we'll see very soon, there is some issue with their p-values. And the ignorance of the pseudotime is only one reason. And we found another reason for that invalues p-values is because that method chooses the number of knots, the degree of freedom, in a data-driven way. But that data-driven way may not have some issue first. And second, it will introduce some uncertainty into the distribution also, because in that, to have that valid asymptotic distribution, the degree of freedom must be fixed beforehand, and it cannot be chosen in a data-driven way. So all these issues together, it actually causes some p-value issue for tracing. But our subsampling and permutation approach can fix that issue. Okay, so for each subsample, what we do is that we fit a GAM to the subsample count matrix and the subsampled and permuted pseudotime. Then we can calculate a value for the test statistic, and these capital B values are the null values of the test statistic SJ. All right, so now if we just want to compute a p-value based on the null values, the simplest approach is to get the empirical p-value. So what we do is that we calculate the percentage of the null values that's greater than or equal to the observed value in our original data, right? And we follow the suggestion by adding 1 to both the numerator and denominator to avoid the zero p-value issue. Or actually not just the zero, but to make the p-value better behave for the FDR. So actually, you can see that there's an issue with this resolution, because if we actually, so let's just think about it, right? So we can see that the smallest p-value we can get out of it is 1 over p plus 1. And it's not actually enough for the FDR control, right? Because if you have too many genes, and if you want to control the FDR to say 1% or 5%, the cutoff on the p-value may be very small. And there, you may not have anything between that cutoff. So basically the cutoff may be bigger than 0, but it's less than 1 over b plus 1. So that will make this empirical p-value low resolution an issue for controlling FDR. Okay, so to address that issue, we provide a fix. And this is a common issue for any permutation test. So we provide a parametric fit to the null distribution to fit those capital B null values. So empirically, we observed that. So we could fit this distribution well using either a gamma distribution, because you can see the original chi-square theoretical distribution is actually a special case of gamma. Or we can fix a 2-component gamma mixture model. So in the original theoretical distribution, it could be a mixture of chi-square, and that's also a special case of a mixture of 2-component gamma. So we will fit each to the parametric, to the observed null values. And then we're going to choose between those two distributions by a likelihood ratio test. And then if we think, okay, pick this distribution or the other distribution, we're going to call the fitted distribution CDF, cumulative distribution function, by fj hat. Don't forget that we're still talking about gene J. So we always carry the sub-index J. And then given this empirical CDF or parametric fitted CDF, we can calculate the p-value as the right tail probability. Okay, so in our package, so basically we implemented this parametric approach, and by default we set capital B to 1000. Okay, but if the user's computational resources is limited, he or she can reduce capital B to 100. And in our paper, we showed that even with B equals 100, this parametric approach still gives quite reasonable p-values. So basically between B equals 100 and 1000, using the parametric approach, the p-values are very well correlated. Okay, so here comes the first set of results by simulation. So we see that here is the expected p-value under the null, because in simulation, we know which genes are not DE. And so their p-values should be following a uniform 0 to 1 distribution. And that's the expected here in the x-axis. Y-axis is the observed p-value. And we can see that, yes, we have a very nice QQ plot here, where I was not saying QQ plot is in a quantile. It's just a scatter plot close to diagonal line. While the existing method TreeSeq has some issues here, Monaco 3 using GLM, it's actually p-values are reasonable, not too off. And these two methods are not designed for single-cell RNA-seq data, but for bulk RNA-seq data. So what we can do is that if your bulk data has a time covariate, it's physical time, and then you can do the regression and see whether a gene's expression changes with time. Although it's not pseudotime, but theoretically it's applicable here. So we just try them here to see how they perform. But unfortunately, the p-values are very off. And one reason we can think for the p-value issue for NB-GAM-seq is that it also uses the GAM. So basically NB-GAM-seq, TreeSeq, and us, we all use GAM. But NB-GAM-seq in the NB-GAM, its dispersion parameter is actually learned from DE-seq2. And we know DE-seq2 is a method designed for bulk RNA-seq, and it uses the shrinkage estimation for the variance parameter to account for the small sample size in RNA-seq. Bulk RNA-seq, we often have like 3 replicates only. So for bulk RNA-seq, given a small sample size, we think that shrinkage estimation is definitely reasonable to make sure the variance estimator is not to having a too large variance. But in single cell, the situation is different, that we have many cells, so the sample size is not small anymore. So in that case, we don't think it's still reasonable to do the shrinkage estimation to obtain a bias estimator just for the sake of reducing variance. And we hypothesize that that might be the issue for the p-value here. But yeah, we don't, that's just our guess, because we didn't look into the code, or we didn't diagnose the issue in detail, but that's our hypothesis here. Okay, so from this result, we see that the 2 bulk methods is definitely not suitable, while TreeSeq and Mono 3 are doing more reasonable job. But we noticed that here, TreeSeq has some I would say inflated p-value issue. So some of the p-values seems to be too big, because otherwise we should have a uniform distribution. Okay, so this is a closer look. We actually perform negative log 10 transform p-values so we can exaggerate the small p-values, because the small p-values are usually of more importance for our decision. And this more clearly shows that, yeah, the two bulk methods are totally off. And as a result, if we compute the false discovery proportion in one realization, and because this is simulation, we know the truth. Yeah, their FDP is just crazy. Although we target 5%, but we are seeing they give a lot of false discoveries. And also, the tree-seq p-values is worse than Monaco 3, worse than ours, and this is also reflected in the FDR control. So tree-seq has FDP 14%, while we target for 5%. So we have more simulation examples in our paper, but this is showing the one example. And also, this is showing that if we don't just target one cutoff, we plot the ROC curve. And it seems that we are still doing the best in terms of the ROC. Okay, and given the 5% control on the FDP, we set a cutoff on that. So basically, we are cutting every method by their actual false discovery proportion, not the nominal, because a nominal doesn't make sense since they are so off. So by the actual FDP, we see that we are having the highest power, while the two bulk methods are doing the worst. So that's why on our real data analysis, we excluded the two bulk methods, but only keep comparison with tree-seq and Monaco 3. Okay, so this is the first real data example. We apply our method to the dendritic cell stimulation by LPS. So it's an immune experiment. Immune cells stimulated by some chemical. So we see that this is the p-value distribution of all genes, right? So we know that some genes must be DE, so their p-values must be small. But other genes, the non-DE ones, are expected to have a uniform distribution. So you can see that that's indeed the case for PseudotimeDE and Monaco 3, but not the case for tree-seq. So this mode towards 1 is no way to be reasonable, because we cannot explain why there are some p-values that are so close to 1, right? So usually this indicates that the method is overly conservative. So you try to inflate some of the p-values, and that will hurt its power. And actually we see that's the case. And between PseudotimeDE and Monaco 3, the difference is that we use GAM and they use GLM. So GLM captures fewer relationships than GAM do. So that's why we would also expect that we have a higher power. So this is the number of DE genes detected by each method and the Venn diagram. So we see that indeed, PseudotimeDE captures most of the DE genes found by tree-seq. Okay, only 36 and 2, right? 38, these are missed, but the majority is captured by PseudotimeDE. And between PseudotimeDE and Monaco 3, there is some discrepancy. So to examine which one is more reasonable, what we did is that we take the difference in pairwise comparisons. So between PseudotimeDE and tree-seq, we look at the DE genes that are uniquely found by PseudotimeDE and the genes uniquely found by tree-seq. And then we perform goal enrichment, gene ontology enrichment analysis. And we found that there are more enriched goal terms in the PseudotimeDE specific genes. And same conclusion for the pairwise comparison between PseudotimeDE and Monaco 3. So on the left-hand side, the pseudotime inference is done by Slingshot. On the right-hand side, the pseudotime inference is done by Monaco 3. But we see that no matter which method we use, the conclusion is actually roughly the same. Okay, so here is some close examination of the DE terms that are in pseudotime specific DE genes, the goal terms. So we see that yes, those terms that are only enriched in the pseudotime specific DE genes are related to immune responses. So they are reasonable. Okay, in the second data set, we performed a similar analysis, And we see that, again, the p-value issue still exists for trait C, just as in the previous example. And still, the pseudotime-specific DE genes are enriched with more gold terms. And this is by slingshot, this is by monocle 3 for the pseudotime inference. And so because this one is for the pancreatic cell development, so we look at the result, and also we see that there are some of the gold terms specifically enriched in the pseudotime-specific DE genes are biologically meaningful. Okay, here are some concrete examples to show a biologically known DE gene and a non-DE gene, what their p-values are. Okay, so let's first look at this one. So for this DE gene that should be caught, so if we look at here, trait C gave a very big p-value, 0.3, so it's going to miss it. But for pseudotime DE and monocle 3, both give it a small p-value. Okay, so from this case, you can see that at least for this data set, the 0 seems to be meaningful because 0 is driving the trend. If you ignore the zeros, just look at non-zero values, the trend is not so obvious. Okay, this is another gene, very interesting. So SST, it should not be called DE, but if we ignore the pseudotime inference, both trait C and monocle 3 give a very small p-value, 0 or 10 to the negative 11. The reason is because of those cells. So those cells right here, they are early in pseudotime and they drive the trend. But if we consider the pseudotime inference uncertainty, as we do, then the p-value is actually bigger. So it's not as small as the p-values given by trait C and monocle 3. So this shows that by considering uncertainty, then the result is less driven by those small number of cells. And the same thing here. If we look at here, so the same gene, and this is the pseudotime by slingshot, this is the pseudotime by monocle 3. So we see that some of the cells are driving the trend to get small p-values. But once we consider the uncertainty, it's less driven by the trend. And also you can see that in this case, the reason monocle 3, although it ignores the pseudotime inference uncertainty, but still its p-value is not as small as trait C. It's just because it uses GAM. So it uses GLM, not GAM. So for GLM, you will not capture this kind of relationship like this, right? Try to fit a linear relationship. So that's why monocle 3 doesn't give up such a small p-value. While we use the GAM to maintain the power, we use pseudotime inference consideration for the uncertainty to correct for those outlier issue, I would say. Yeah, this is the second example. And the third example is a bifurcation trajectory. So we see the trajectory goes this way and it goes this way. Okay, so as we can see, in some cases, we would infer a trifurcation instead of a bifurcation. And actually, biologically, it's hard to say which one's more reasonable because if we consider this trifurcation, there's also some explanation for the third trajectory. So we leave out this controversy. But in our method, what we do is that if we use a pseudotime inference method, which is our method is flexible, adaptive too. So use whatever method. So if the original data gave us a bifurcation trajectory, we only pick the subsamples that also gave us bifurcation trajectory so that we can have the same topology between subsample and the original data to make our inference easier. Otherwise, if the subsample has trifurcation trajectories, we will have some issue about matching the lineages, like which trajectory we should match between here and here. And that actually complicates the issue. Okay, so we only keep the subsamples that maintain the topology. That's the message. And then we actually look at the DE genes again, and we look at the functional impact. So for this one, we perform the gene set enrichment analysis to see how many gene sets are enriched in the DE genes found by each method. So this analysis, again, emphasizes the importance of valid p-values. So we see that because in the GSEA, gene set enrichment analysis, we need to input a rank to our DE genes, right? And in that rank list, each DE gene is going to have a ranking score. And here we use the ranking score as the negative log 10 p-value. which is what people often use. The smaller the p-value, the more top the gene should be placed. But the p-values, if it's not really distinctive, then we see that, okay, the trace-seq p-values, doing it this way, will give us very small or almost non-enrichment of the pathways, the significant gene set, while our p-values give us many pathways. So I think this actually also illustrates the importance of using p-values on the correct scale, not just the ranking of genes, but the p-values, nominal values are very important. Okay, so we have two more examples in our bioRxiv paper, but I will just want to stop the real data part here because we have another thing to talk about. So if you want to learn more about the generalized additive models, there is a book written by Dr. Simon Woods, and he also is the author of the R package and GCD for the analysis, and we use this package in our PseudotimeDE method. So if you want to look into that, here are the resources. Okay, so that's all I want to say for the first part. Do we have questions before I move on to the second part?
So I have one related question, which is, I could imagine that these kinds of issues would also arise if you're just doing differential expression analysis based on clusters that are inferred from data. So not, I mean, so any kind of latent structure that is inferred from the data. So
Exactly.
have you looked at these kinds of issues in the context of just differential expression analysis applied to clustering?
Yeah. Actually, we haven't yet, but I think, yeah, you made a very good point, valid point. It's definitely an issue. And I think, yeah, it can be easily generalized to that. Our idea can be easily generalized to that problem. And also the same issue, right? What clustering algorithm you will use is the same as what pseudotime inference method you will use here. Yeah, definitely. It's something, I think it's definitely something we try to do. But indeed, I think so my student indeed find that there are some issues with the application. So a similar message to the bulk method issue here as related. So we also saw that if people try to apply the bulk method to single cell RNA data, there could be some issue with the p-values, just because, as I said, the shrinkage estimator for the variance may not be reasonable anymore.
Yeah, I think, yeah, that's, yeah. I think it's really nice to think through all of the uncertainties in these analyses.
Yeah.
I guess another question is, there is this whole kind of statistical literature on post-selection inference.
Oh yeah, exactly. Yeah.
And is any so I guess, I mean, one question is any of the things that you're thinking about, are those applicable more broadly to the kind of problems that people think about in the post-selection inference literature?
Yeah,
and,
actually,
and the other way around, are there ideas from the post-selection literature that could be useful in this context?
I think that's a very good question. And actually, we mentioned this in the discussion part of our paper. So I think the whole problem is very related to the post-selection inference. That is, some of the model tuning parameters are chosen based on the same set of data, and then you're doing inference on the data after fixing those parameters. So I think the whole idea is very generalizable. But I feel like the term post-selection inference literature is still not directly applicable to our case, because I think they focuses on the cases where they should still be able to derive some valid null distribution for the test statistic in post-selection inference. But I think our problem here is a little too complicated to actually do that. So yeah, so we still think we haven't found a concrete link as a solution yet. But hypothetically, I would say that's something we should consider.
Yeah, it seems like that literature is primarily focused on getting asymptotic distributions, and for that you need a lot of assumptions and fairly simple models. So presumably that's not directly applicable here.
Yeah, exactly. Especially, I think pseudotime inference itself is quite complicated problem. That perspective, yeah. Thank you, Shram. That's a very good question.
Hi, Professor Lee. I have a question. I remember you mentioned one part when you want to choose a distribution between a gamma distribution and a mixture of gamma distribution, and you compare the likelihood. But you have a hyperparameter gamma, so I wonder how are you going to compute this gamma?
Oh, so when we fit the mixture model to our data, the gamma will be estimated. So we're going to estimate gamma alpha 1, beta 1, alpha 2, beta 2. So this is a 5-parameter model, and this is a 2-parameter model. So when we do the likelihood ratio test, we are actually considering this gamma distribution to be nested under the 2-component gamma. So that's why we're doing the likelihood ratio test.
Oh, I see. So gamma is not a hyperparameter. Okay, I see.
No, it's going to be fixed to be fitted from data.
Okay, thank you.
Yeah. Thank you. Okay, thank you. And then I guess I can move on to the second part, scDesign2. So in the single-cell RNA-seq field, many experimental protocols have been developed in the recent 10 years. You can see the timeline here. More and more protocols emerge, right? And as the technologies improve, the newer technologies can sequence more cells in one experiment, but there is no free lunch. So usually for the technologies that can sequence more cells, they sequence each cell in a more shallow way compared to technologies that can sequence fewer cells, but can sequence each cell in the deeper way. So there's a trade-off between the depth and the breadth of the result of the measurements. Okay, and also there are tag-based versus full-length protocols. So for example, 10x Genomics is tag-based, while SmartSeq2 is full-length. So what it means is that in 10x Genomics, every RNA molecule you only sequence its 3' end. And in SmartSeq2, you're going to sequence fragments from the full-length RNA molecule. So they give you different information. If you're only interested in counting genes in a cell, then you need to decide which protocol you want to use, right? So I think the protocol choice is part of the experimental design. And also, given the protocol, how would you determine the number of cells to sequence and the total sequencing depth? So the sequencing depth will determine your budget, will be based on your budget, and optimal cell number will also reflect some breadth and depth trade-off. So the more cells you sequence, the shallower each cell you will get, and vice versa. So basically, these issues are all something we need to set as experimental parameters. So how to find the optimal design? We think that's a computational question. So this diagram shows that, okay, let's say this is the true gene expression matrix. We have 3 cell types, and you see that, so because it's a simulation, so these genes are highly expressed in cell type 1, these highly expressed in cell type 2, these highly expressed in cell type 3. So if we only sequence 50 cells, we can see those genes clearly, right? So we have the same number of genes, but only 50 columns for the 50 cells. If we sequence 2,000 cells, we get more cells, but the distinction may be not as much, right? The genes are not so clear as here. So basically, we need to have that trade-off to find the best trade-off between the breadth and depth if our total number of reads is fixed. Okay, that's a design question. Another important question is to benchmark computational method. So here from a paper, we obtained the cell level and gene level analysis. Those are typically performed on single-cell RNA-seq data. So on the cell level, we have cluster analysis, so to divide cells into clusters, and the trajectory analysis, which we just saw. And so based on the clusters and the trajectories, we can also perform some analysis like the clustering annotation, and also we can look at the gene expression dynamics. That's something we just saw, pseudotime. On the gene level, we can look at which genes are differentially expressed between 2 clusters, and we can perform gene set enrichment analysis and go-term analysis, and we can also look at gene regulatory networks. So it seems that for many tasks, and each task will require some computational method. And because of the popularity of the field, we often have tens of thousands, not thousands, dozens of methods for each analysis, and even more, maybe hundreds for some popular analysis like clustering. So if you check there's a website called SC Tools, you'll see that the number of computational tools grows sharply in recent years. So if for experimental users, data analysts, it's very important for them to pick a reasonable method for their analysis. So yeah, so here are some examples. Like for dimensionality reduction, we have these methods: clustering, rare cell type detection. If you want to detect a small cell cluster that may be missed by global clustering, you may want to apply this rare cell type detection method. Trajectory inference method we have seen already, and the identification of EE genes between cell types. So each task has many methods available, but how do we choose one? So our motivation to develop scDesign2 as a simulator is to address both experimental design and computational benchmarking questions. So our hope is that with a realistic simulator, its synthetic data carries ground truth, then we can validate the experimental design, right? We can evaluate the effectiveness of design, and also we can benchmark or compare methods for the same task. Okay, so this is the philosophy. We want to use real data to fit a model, and we want to use the model to generate synthetic data. And the hope is that the synthetic data would capture key characteristics of real data. So this will make us feel, yeah, the model is trustworthy. So we can use synthetic data to make some conclusions. Okay, so there are many simulators that are out there. So here's a summary in our paper. So we try to evaluate each simulator from 6 perspectives: whether the protocol is adaptive to different whether the simulator is adaptive to different experimental protocols, whether the simulator preserves genes. So what do we mean? So in some simulator, its synthetic data doesn't carry genes, which means that if we call genes as the rows, then you cannot match the rows in the synthetic count matrix to the rows in the real matrix that used to be trained the simulator. So basically, you cannot have that matching of genes to genes. And does it capture gene-gene correlation? That's the major motivation of our development, because the existing simulators, most of them actually do not capture gene correlations at all, or they do not explicitly capture gene correlation. Although we hypothesize that given their approach, they may implicitly capture gene correlation. And these include ZINB-GAM, sparse ZIM, curse-black, and scGAM. But we are going to check it, okay, because they don't say we capture gene correlations explicitly. And another important characteristic is whether the protocol can allow flexible cell number and sequencing depth. So can you change the number of cells in the synthetic data? Can you change total sequencing depth in the synthetic data? Unfortunately, many of them cannot. So for example, ZINB-GAM. So although we know ZINB-GAM is primarily used as a dimension reduction method, but its model can be used to generate synthetic data. If you use your model in that way, the limitation is that you must keep the same number of cells as in the original data. And that's a very big restriction. It also doesn't allow you to tune the total sequencing depth. I think it's reasonable because it's not primarily designed as a simulator. So I will not take that as a criticism, but I will say that it cannot be used for this purpose. And whether the model is easy to interpret. So this is a quite subjective judgment, but we think scGAM based on deep neural network is not so easy to interpret. And some of the methods has too many parameters, so not so easy to interpret. So I would say this is more like a subjective decision. Okay, finally, whether it's computationally efficient, how fast can you train it, or how fast can you use it to generate samples, new data, and whether it's sample efficient, can they be trained on a small number of cells? So for these 2 considerations, scGAM is not very good because of its deep learning nature. All right, so we want to propose our scDesign2 to satisfy all the 6 properties, especially capturing gene correlation. So why do gene correlations matter? Here is a toy example showing that for two genes, this is one gene has this marginal distribution, okay, almost same marginal distribution. Another gene having the same marginal distribution. But the two genes could be positively correlated, not correlated, or negatively correlated. So you see that the correlation is not reflected in their marginal distribution. But this correlation could be biologically important because usually we will use the correlation to interpret some hypothetical regulatory relationships between two genes. But if our simulator doesn't capture that, then the simulator cannot be used for some multi-gene analysis benchmarking, such as this gene regulatory network construction, right? Since your synthetic data already missed the gene correlation, this analysis will become nonsense on the synthetic data. Okay, so this is the diagram of the scDesign2. Again, I will talk about the details in a step-by-step next time, the next slide. But here, we want to just show you the big idea. So scDesign will fit a model, generative probabilistic model to each cell type. So let's say we have a capital K cell types, and these can be predefined or can be obtained by clustering. We'll talk about this issue later. So for each cell type, we get its matrix, the matrix. So here the cells are the columns, the rows are the genes. For each submatrix, we're going to fit a joint distribution for many genes together. So in practice, in our model, we fit thousands of genes. We didn't fit all the 20,000 genes for human cells. The reason is that many genes are not expressed or too lowly expressed. So it doesn't make sense to really include them because but we just don't have much information to learn about those genes from real data. And also that dimensionality will make this model fitting more difficult. So we must restrain our number of genes, but still it's hundreds to thousands, so that we're capturing those important genes joint distribution. And given the joint distribution, we are going to allow user-specified sequencing depth and number of cells to generate synthetic cells. So this is a case where we make the sequencing depth quite low. So you see the synthetic cells look like this, while the real cells look like this. So we are showing the effect of sequencing depth. And then we'll use the synthetic cells to guide experimental design or to evaluate computational method. Okay, so now notations. So for this set of my slide, we use the CAM matrix. We call the CAM matrix of X and the P genes as the rows and cells as the columns. We assume that it contains cell types and the cell membership, with which cell is in which type is already known in advance. So we assume that for each cell type K, we have the number of cells as NK, and we have the corresponding sub-CAM matrix as XK. So our goal is to fit a parametric model for all genes expression for each cell in cell type K. So this all, I should quote it, so it means that the important genes we want to capture. Then for the uninformative genes that are constantly off, we just remove them. And for the simplicity of notations, from now on, we're not going to keep the sub-index, it should be superscript K, because here we are going to focus on one cell type only for the model fitting. Okay, so regarding the cell type definition, right, that's a very practical question. Even though the real data may have cell types labeled, but do we think the cell types are refined? So to check that, we actually leverage a recent method paper called Rogue Score to check the so-called homogeneity of each cell cluster. So for example, STEM is a cell type in real data, but using a Rogue Score, basically it's checking how well the genes in each cluster follows the negative binomial distribution. Because for the UMI-based data, negative binomial distribution is something people think is reasonable. So if the one gene has distribution that cannot be well explained by negative binomial, then we will divide this type into a subcluster and then calculate the Rogue Score for a subcluster. So basically, we see the Rogue Score should always increase as the number of subclusters increase. But we want to look for the plateau point, the tipping point, the change point, so that we stop here. We don't do further division. So clearly see that for the same cell type, although it's labeled as one type in the real data, we found it's more reasonable to be divided into two subclusters, and also for goblet. Yeah, so basically what we did is that, although I haven't presented the method yet, but we can see that indeed, using the cell type refinement, we can actually have synthetic data that mix better with real data than using the original cell types. So clearly for the goblet, which is the brown color here, you see that if we don't do the refinement, the synthetic data will give you one cluster in the TC visualization, while the original data have two clusters, one big, one small. And with the refinement, now we can capture that property. So basically, this is showing the original data, and this is the data for training simulator. This is the unused data as test data for evaluation. This is the synthetic data with refinement from our method. This is the synthetic data without refinement. And for each set of synthetic data, what we do is that we mix it with test data to do joint visualization. So in the joint visualization, red is synthetic data, black is test data. So the better they mix together, the higher this value will be. So this is called a MILISI value. So it's an integration index to indicate how well those two data sets mix together. And in some method that does data integration, like Harmony, it uses index to show how well the integration works. And without the refinement, we see the index is lower. After refinement, it's bigger. Okay, so this is just to address the cell type definition issue. With the cell types defined, so our approach to fit a model to the data has two steps. One is to fit a marginal distribution for each gene J. And we use the gene counts, okay? So let's say this is the cell J's vector for gene 1 to P. And these are counts. And we are assuming that these vectors are IID for cells. So in other words, we assume independence of cells. Each cell is an independent draw from a joint distribution. And then for gene I, we consider Xij to come from a gene-specific distribution. So here we consider zero-inflated negative binomial as the distribution. So it has the gene-specific proportion parameter for being on, and the mu I is the gene-specific mean, psi I is the gene-specific dispersion. So we know that the ZINB is a general model that includes NB for sure, and Poisson, and zero-inflated Poisson. So basically, if we set this dispersion psi I to infinity, this model is reduced to zero-inflated Poisson. And if we set Pi equal to 1, then we change it to NB. And if we set Pi equal to 1, psi equal to infinity, we reduce it to Poisson. So that's why this is the general model that includes all the 4 cases. And please note that similar to what we have seen before, the latent variable Zij indicating the gene to be on or off is not observed. Okay, so given that count matrix with P genes and N cells, our first decision is whether we want to go for Poisson or NB. So our decision is very simple. We calculate for gene I its average across cells and its variance across cells. So this is the sample mean, sample variance, and we compare the two. Because we know that NB is used to account for the over-dispersion issue that Poisson cannot address, right? Poisson requires mean to be equal to variance. But if the variance is much bigger than the mean, the Poisson cannot be well fit to the data. So we use NB. So our simple judgment here is basically, if the variance is not bigger than the mean, we just fit Poisson or zero-inflated Poisson, and we will decide between those two using a likelihood ratio test. Otherwise, if the variance is bigger than the mean, we fit an NB and ZINB, and then also choose between the two using a likelihood ratio test. And then for the p-value, we set on the likelihood ratio test, we use the 0.05, so that we will choose between whether using zero inflation or not using zero inflation. So admittedly, actually, when our paper were under review, a reviewer raised this issue to say, why would you choose the model? Why don't you just fit ZINB to the data? And that's it, because the ZINB is the most general model. And our answer to that is this choice, this model choice step is actually can provide us with more information about the data characteristics. And that data characteristics may be meaningful, may be biologically useful. So we will see that in a later application of our method. But for now, this is just to choose a count distribution for each gene I. Then how about the joint distribution? This is the most challenging part, not tackled by previous method. To tackle this issue, we use the copula framework. And here I'm going to introduce it. So the copula here is a CDF that will map from the p-dimensional gene expression space to 0 to 1. So it's a CDF, joint CDF. And this is the key of copula. It's called a scalar theorem. And this scalar theorem is about the existence of a copula. And this copula itself is mapping from the p-dimensional 0 to 1 to 0 to 1 interval. So from a cube to an interval, it will satisfy that the joint CDF can be written as this C with its p arguments as the marginal CDF of the p variables. So basically, it's a way to compose a joint distribution from p marginal distributions. Yeah, so this theorem says that this function C is unique for continuous distributions, but not unique for discrete distributions. So in other words, although there exists C, but the C is not unique for discrete random variables. And that's our case. Okay, we are having discrete random variables because we model each gene's expression in cell as a count. Okay, so to address that issue, we use the technique called distributional transform to make a discrete random variable become continuous. So what we do is that we will randomly sample a variable Vij from uniform 0 to 1 as a weight. And then we're going to create a continuous random variable. And this continuous random variable is going to be between 0 and 1 by combining the CDF of Xij minus 1 and the CDF of Xij, take their weighted sum. The continuity comes from the fact that Vij is continuous. It's drawn from uniform 0 to 1. So this is how it works. So essentially, if you look at a CDF, and this is the PDF, so the random variable is Poisson, so it's discrete. So this is the probability mass function of the Poisson at the values it takes with non-zero probability mass from 0 to 7. This is the corresponding CDF. So you can see it's a step function. And the distributional transform is trying to smooth out the CDF, and it becomes this. And what it essentially does is that it will actually uniformly distribute the probability mass, say at 0 to the interval from 0 to 1. So you have a uniform density here. You will also distribute the probability mass at 1 uniform to 1 to 2. So you will have a uniform density here. So with this, you're essentially converting a discrete random variable to a continuous random variable. And that's the CDF. Okay, so with this transformation, so every gene I in cell J, Xij, is going to be converted to Uij. It's a random variable between 0 to 1. Okay, and then what we did is that we will make each Uij as Gaussian. So what we did here is that we applied this is the CDF of a standard normal distribution, standard Gaussian. We apply the CDF inverse. So we're essentially transforming each Uij to a Gaussian variable. And then we're going to fit, this is called Gaussian copula, because the joint distribution is going to be described by the correlation matrix. And this is equal to covariance matrices here in this joint p-dimensional Gaussian. We have 0 mean and unit variances. So then the correlation matrix is just the covariance matrix. And using this, we are going to have a joint distribution. So in other words, we're trying to specify the joint distribution of P genes using the Gaussian copula. And in the idea of the Gaussian copula is that on the transformed random variables, so from the original counts to Gaussian-like variables, we're going to capture the correlation on the transformed variables. So please note that the correlation matrix is for the Gaussian, it's not for the original counts. But there is some interesting relationship between the original gene correlation and the Gaussian copula correlation. So because the copula is essentially based on ranks, right? So when we see that we do this transformation first from the original count to its CDF, and then from its CDF to the Gaussian inverse CDF. So we're essentially only keeping the rank or the quantile. So that's why the correlation for gene is not for the Pearson correlation, it's for the Kendall's tau, which is the rank-based correlation. So this is an interesting relationship saying that given the Kendall's tau of gene H and gene L, you can actually write this Gaussian copula correlation via the spline transformation. So this shows that our use of Gaussian copula can indeed provide some interpretation of two genes Kendall's tau correlation. And this also suggests that if you want to estimate the Gaussian copula correlation, you could actually you can calculate the Kendall's tau between two genes and insert it here, and then you can calculate the Gaussian copula correlation. However, we don't recommend this approach because if you do that pairwise for genes, the resulting correlation matrix may not always be positive semi-definite, and that would be not ideal. Now that will cause issues for data generation because that's a requirement for this correlation matrix. So that's why we don't do this. Instead, what we do is as follows. So we are basically treating the marginal fitting and the joint marginal estimation and the joint distribution estimation as two steps. So we first fit the marginal parameters for gene I, so the parameters for the ZINB distribution. And we denote the corresponding CDF as Fi hat. And then we apply this distributional transform using the fitted marginal, okay, to create a UIJ. And finally, we take the Gaussian inverse to get a vector, p-dimensional vector for each cell J. And using those J from 1 to n, using those vectors, we will estimate the sample correlation matrix for the Gaussian copula. So it's a two-step approach estimation. Okay, so one question which comes out is the high dimensionality. We know that there are many genes. So then how can our estimation do? So as we said, we actually rank genes by their mean expression. So the higher the expression, the more top the rank is, right? So we see how about we can include top 100 genes, 200 genes to 2000 genes, and how the estimation accuracy would be. So for this one, we are basically using simulation to capture the idea. So what we did is that from real data, we estimate R hat, and then we use this fitted model to generate data with different number of cells. That's what we call sample size. 5 cells, 10 cells, 20 cells, 50 cells, 100 cells. And then we estimate the matrix R again on the synthetic data. And then we evaluate that matrix to the matrix we use for generation by the MSE, mean squared error. So as expected, we see that as the number of genes we increase, the estimation becomes worse, right? The larger the MSE, the worse. However, we see that if we just have 100 genes included, then even a sample size of 10 cells, our estimation is not so bad. So basically 0.09 means that on average, the correlations differ by 0.3. 0.04 means that on average, correlations differ by 0.2. And 0.01 means that on average, correlations differ by 0.1. So we see that the estimation accuracy is not so bad. And if we have 100 cells, we can actually do pretty well even for estimating 2,000 genes. Yeah, I think that the reason is why the dimensionality is not so much of a concern here. It's just because many genes are not expressed and many genes have very low correlation. So our correlation matrix is not very dense in the beginning. So actually that sparsity helps with this estimation problem. So it's not as bad as we thought it would be. Okay. And also on the left-hand side, we fix the number of genes and change the sample size. So we can see how many cells we need to achieve reasonable accuracy. Again, with 100 cells, we can do reasonably well up to 2,000 genes. Okay, then finally, with the model fitted, the joint probability model using Gaussian copula, we need to do data simulation. For this part, I'm going to skip the technical details, but basically we need to input the model parameters we learned, we estimated, and the cell type proportions also from real data, number of cells to simulate, and the total number of reads to simulate. These are user-specified parameters. So first of all, given the number of cells to simulate and the proportions, we will determine the number of cells for each cell type. And then we will determine a scaling factor about the number of reads. Okay, so because we fed the model to the original data, but the number of reads may be quite different from the synthetic data, given the user suggested input parameter. So we need to take that difference, the different scale into account into the scaling parameter. And then we'll use the scaling parameter to scale the mean of our marginal distributions, like the Poisson or negative binomial. And then we're going to draw data, generate data from the Gaussian copula, via the Gaussian copula, and for each cell type, so to obtain the synthetic data matrix. So it's basically the reverse of what we fit the model. All right, so now let's look at some results. So the first result is to compare our method with existing method. So these methods, many of them do not capture gene correlations at all, including our previous method, scDesign. So that's why we call this scDesign2, because in the previous method, we also want to develop a simulator, but back then, the simulator doesn't capture gene correlations. And that's why we're motivated to improve it as scDesign2. So just for comparison, we also can have scDesign2 without a copula component. So we don't use copula to capture gene correlations, but just the marginal distribution. And we see that, yeah, we cannot capture Pearson correlation or Kendall's tau at all. So this volume plot showing the distribution of Kendall's tau across gene pairs in real data. So while some methods like scGAM can implicitly capture gene correlation, but we see that it's not really close to real data and has a much longer tail here. Well, and this means that it captures some negative correlations not in the real data. And also ZINB-GAM and sparse-ZIN, they have some issues to capture the negative correlation. So probably this can give you a better sense, the heat map. So this is the heat map of the gene-gene correlation matrix. We see this is the training data used for training simulators. This is test data. And this is the heat map output by scDesign2 synthetic data without copula, no correlations at all, ZINB-GAM and sparse-ZIN. So we see that our heat map most resemble that of the real data. And that's for one data set, one protocol. So this is 10x genomics, and this is for SmartSeq2. Again, the same conclusion holds visually. All right, so now how about, then these heat maps for one cell type together, by the way. This is DC1, this is goblet. Now, if we mix cell types together, then how does it look? Training data, test data, synthetic data by scDesign2, ZINB-GAM, sparse-ZIN without copula. So in this joint visualization plot, joint dimension reduction, we can see very well that whether the synthetic data mixed well with real data. And please note that this is before we do the cell type refinement. So that all the simulators are using the same original cell types. Still, our synthetic data makes much better with real data than other simulators do. That's the TC, and this is the PCA. Same data, different visualization, and the same conclusion holds. Okay, now application one.
So we want to argue that our simulator is not just applicable to single-cell RNA-seq data, but also other single-cell technologies, as long as the data are counts. So here we show that for the merfish data, the spatial data, please note that here we didn't aim to generate spatial coordinates, just the counts. Still, for the synthetic counts, it mixed well with the test data as before. So better than other simulators do. This is for merfish. And this is for another spatial technology called PCIC. So we're still doing the best in terms of the mixing. All right. So next step is to show that, yes, the model selection for marginal distribution is meaningful. So this is the benchmark data set of blood cells in the benchmark paper generated by multiple experimental protocols. Here we pick 3, 10x genomics, DropSeq, SmartSeq2, and the same genes. Okay, so we want to show that the model choice in our scDesign2 reflects interesting results. So first of all, we can use the model selection to say how many genes are selected to be zero inflated, how many genes are not. And clearly we see for 10x genomics and DropSeq, almost none genes are selected to be zero inflated. While for SmartSeq2, half of the genes are selected to be zero inflated. So you can see that this is one case, this is another case where zero inflated models are chosen over non-zero inflated models. And also we can see that from this histogram, we can see how well the theoretical marginal distribution fit to the data. So clearly for the for the 10x genomics and DropSeq, we see much better fit than for SmartSeq2. And a possible reason for the zero inflation of SmartSeq2 is the PCR amplification, because it doesn't use the unique molecular identifier to remove PCR duplicates, while 10x genomics and DropSeq don't have that issue. Okay, finally, for the experimental design and computational benchmarking, this is an example for cell clustering. So we generate synthetic data for different number of sequencing depth. So the total number of UMIs increase. And we're showing 4 examples here corresponding to these 4 dots down here. And for each synthetic data set, we apply SURAT and SC3 as 2 popular clustering methods. And this is the true label used for data generation. So based on the true labels and the cluster labels, we can calculate 2 accuracy measures for clustering: the adjusted mutual information and adjusted rand index. And we can see that for both methods, as we expect that the accuracy increases as the total sequencing depth increases. However, there is some saturation effect, right? If you go beyond the sequencing depth, then further increase doesn't help that much. And we see that this dashed line is marking the real data sequencing depth. So what this suggests is that by our simulator, if we increase the sequencing depth further, we could improve the clustering accuracy. Okay, that's the first message for experimental design. And then between the 2 clustering methods, we see that SC3 is doing better when your sequencing depth is high. However, SURAT is more robust for low sequencing depth, because for some cases, SURAT can still output clusters, while SC3 just cannot output anything. So yeah, that's just the robustness evaluation. Okay, on the right-hand side, we fixed the sequencing depth, but changed the cell number. And we are actually showing the cluster result, SURAT, SC3, and true cell types. So for this, we will expect a first increasing and then decreasing trend. The reason is that if there are too few cells, the clustering can be different. That can be difficult. While there are too many cells, then each cell is not measured accurately. The clustering result will also decrease. So we see this trend for both methods. And actually, interestingly, the result shows that for SC3, it works better when you have fewer cells. But again, SC3 doesn't cover a wide range as the SURAT, because SURAT is more robust. So when the cell number is super large, SURAT can still output something. And also we see that the cell number in the real data doesn't seem to be optimal for either SURAT or SC3. We could actually sequence a slightly fewer number of cells, but still to achieve a better accuracy. That's for clustering. And this is for rare cell type detection, another task. For this one, we compare two popular methods, FIRE and GenieClus2. So these two methods use different methodologies. So FIRE uses outlier detection to detect rare cell type, while GenieClus2 is based on clustering. So you will first divide cells into clusters and then call the smallest cluster as a rare cell type. But sometimes you see GenieClus2, although for some cases, when the sequencing depth is high, it's overall doing better. But we see a special case when it completely failed, and that's here. So from here, we can see the issue is that it mistakenly called another cluster to be rare cell type, but missed the true rare cell type. So it just called this one as rare cell type instead of this one. So it gives a large false positive and also false negative. And that explains the biggest drop here. While compared to GenieClus2, FIRE is more robust. We don't see this weird trend. And also, we can see that right here, FIRE can work for a cell number very big, while the GenieClus2 cannot, because again, it's limited by its clustering property. If the clustering cannot be performed when cells are too many, then it cannot detect the rare cell type. Yeah, so basically, we just show that by changing the sequencing depth and by changing the cell number, so fixing the sequencing depth, change cell number, here is fixing cell number, change sequencing depth, we can evaluate the performance of computational method in this whole spectrum. So users can have better idea about the robustness and the stability of the method. And also we can use this diagram to show the synthetic data, and we can decide a reasonable number of cells and the sequencing depth to use for future experiments. So we can learn from pilot data or public data. So to summarize this part, I introduced our scDesign2 as an interpretable simulator because of use of probabilistic model to generate realistic single cell gene expression count data with gene correlations captured. It was motivated by our previous work at ZINB-GAM, but that one cannot capture gene correlations, but this one can. And it uses a multi-gene generative model. It's probabilistic, transparent, and interpretable. And it can guide experimental design and computational benchmarking. And so for future work, we want to extend the current model to account for continuous cell trajectories. Just as the first part we said, right now it can only simulate data for discrete cell types, and that's the biggest limitation it currently has. So yeah, so I want to say that, sorry, I think I missed one slide, but probably because this version. Yeah, I want to say that if you want to learn more about the copulas, you can check out a book called Introduction to Copulas by Dr. Roger Nelson. So that book is not very thick and it gives a very good introduction to copulas. So finally, I want to thank my student Dongyuan, who was here for the first part, the PseudotimeDE, and Tianyi, who is also here for the second part at ZINB-GAM2, and also Dongyuan contributed that other spatial data technology result to Tianyi's paper as well. And also my former student Vivian, who developed scDesign and who also participated in this second scDesign2 development. And also I want to thank my funding agency. Thank you.
Thanks so much, Jessica, for that great talk.
Thank you. Hopefully it doesn't take too long. It's longer than I expected because I explained some of the details.
Well, I think we have also lots of questions. So yeah, I think that was great. And yeah, I also, yeah, I think the copulas idea was particularly interesting. So for those of you who are hearing about this for the first time, I think the key kind of takeaway is you want to simulate a joint distribution with known marginals. And copulas kind of give you a mathematical framework for doing that. So yeah, highly encourage people to read up on that. Any other questions for Jessica and Tianyi and Dongyuan?
And also, Sharon, after I fix in some of the typos, I can send you the slide if the students want to have a copy.
Yeah, that would be great. Thank you.
Hi, Professor Lee. I have a question regarding the part when you try to transfer the x to some continuous function, continuous variable u. So that part, if I understand correctly, the reason why we do that is because you want to find a unique.
Yeah, exactly. Yeah, because of the identifiability, we want to make the random variable continuous.
So the thing I'm concerned is when you use a function to transfer from x to u, is that function unique, or is the way you produce u is unique? Because ask this question is because yeah.
No, it's not. As you said, because the Vij are random draw, right? So these are random. So basically Uij. So by basically changing the seed, random seed, when you repeat this, then the Uij will be different because the Vij. Well, actually, we show that despite of the randomness introduced in this step, we actually have pretty stable result. Okay, that's I want to say that this is in your current implementation. But theoretically, we don't have to do this. So for example, if you think about this way, if we just uniformly assign the probability mass to the interval, then this is a deterministic transformation. So we could do this in a deterministic way. It's just that for simplicity, we use the random sampling from uniform 0 to 1 to achieve this. And we show that it's robust. Yeah, I want to clarify.
I see. So another following up question is this function, so one randomness come from the random seed generated from the uniform distribution, and the other randomness I'm considering is there possible that there are some other valid function that can transfer x into u without using this specific form? Yeah,
I think definitely. This is just one way. This is just one way to, because essentially we want to get x, because essentially we want to get is the this one. If you look at the original copula, we want to get this one, f(1x1j), right? We want to get this, and we hope this f is continuous. So this continuous transformation just to make the random variable CDF from the stepwise discrete CDF to the continuous CDF. But this is not the only way. So for example, what we do is that we distribute the value, like say 0, the probability mass 0, uniform to 0 to 1. Then you can also do it backwards, right? You could distribute it to the interval -0 to 1.
Yeah, exactly. Yeah. So I'm thinking, what's your intuition if we change the transformation function from x to u? How would that influence the empirical result?
I don't know. So we haven't tried other transform, but my hypothesis is that it shouldn't be too sensitive to that, because essentially your data generation is also following the same. Yeah, the data generation. I wouldn't say that. So for the data generation, what we do is that we generate Gaussian vectors first, and then marginally, we will transform the Gaussian back to the marginal CDF. And the transformation is just using the quantile. So yeah, I think that I will leave that as an open question, because we didn't try other transforms. Yeah.
Thank you. Thank you very much.
Great. Any other questions? All right. So I'm going to stop recording in that case. And